Hi everyone, Brenda Gustin here. And in this unit, we're going to talk about microbial genetics. I'm going to start off in chapter 8. And I just want to give you a heads up that there is a lot more information about microbial genetics in this chapter than what I will be covering. My objective for this topic for this course is to remind you of some of the basic foundational things that you should have learned in other courses about genetics or about DNA, RNA, gene expression kinds of things. And then how do those concepts play out in bacteria and viruses? How are they unique? So this is not intended to be a complete, thorough restatement of everything about DNA replication, transcription, translation, all of that. You should have gotten that in another class. And having a really rudimentary or really basic understanding of that material is needed to understand what I'm talking about, but you don't necessarily need to understand all the details of that. I'm going to be going back and forth between the book and some diagrams that I put in the lecture study guide. And I'll give you an alert when I'm switching between things. So here I am in section 8.1, and your learning outcomes say define the terms chromosome, genome, gene, and compare these in eukaryotic cells and prokaryotic cells and viruses. I actually have a practice question where you do a matching for that vocabulary. So I actually have that vocabulary, those words defined in the lecture study guide under the practice questions. But I just want to kind of review where you can find it here and kind of review these concepts. So here it's talking about the genome is the sum total of genetic material in an organism. Remember that eukaryotic cells and prokaryotic cells have DNA as for their chromosomes, so DNA makes up the genomes. In some bacteria and fungi, there actually are extra pieces of DNA called plasmids, and we mentioned these earlier this semester. And these are what we call extra chromosomal pieces of DNA outside of the bacterial or the fungal chromosome that have genes on them that give the cells extra extra proteins that give them extra abilities. The reason why I cover plasmids in this class is because we're going to be talking about antibiotic resistance, and a lot of times bacteria can pick up these pieces of DNA in the form of plasmids that have antibiotic resistance genes on them. So here's an example of a bacterial cell, and here is the bacterial chromosome, but these pieces of DNA outside of that chromosome are called plasmids. And they just have a couple genes on them. Remember that viruses don't just don't necessarily have DNA as their genome. They can have DNA or RNA depending on if it's a DNA virus or an RNA virus. And that's going to be an important concept in this chapter. Some other things that distinguish these three different types of microorganisms is the chromosomes are distinctly different. Remember, eukaryotic chromosomes are inside of a nucleus. They're the pieces of DNA. And so depends on the organism, but human cells have 46 pieces of DNA or chromosomes in the nucleus, whereas most bacterial cells just have one circular chromosome as opposed to 46 linear chromosomes. Most viruses have one chromosome, but there's actually a lot of variation there. And so they're going to have one or more pieces of DNA or RNA in their genome. Let's see, we mentioned the word gene. A gene can be defined. Oh, there's lots of actually different definitions of the genes. The definition I use for a gene is it's enough DNA or RNA to enough DNA. Let's talk about bacteria. To encode for a protein or to encode for a group of similar proteins. So a gene's enough DNA or enough genetic material to encode for, to have the instructions to make a protein or to make several similar proteins. Here it gives you, this discussion just tries to compare what some of those sound like, some of the differences in these organisms. So genomes can vary greatly in size. viruses have 4 or 5 genes, can have 4 or 5 genes, the smallest ones. And one of your learning outcomes was explain why viruses can function with small genomes. Uh, they they only need a handful of genes because remember the virus is living inside of a host cell. Think about kids living with their parents. They don't need as much money. They don't need their own couch. They don't need their own dishes. They use what the parents have. So viruses are kind of the same way. They don't need to make all the proteins that are necessary for existence. They just need to make a few that are unique to the virus. So because viruses have a host cell in which they live, they need relatively few genes. Here it tells you the bacterium E. coli has 1 chromosome and about a little over 4,000 genes. Compare that to a human cell that has 46 chromosomes, 23 pairs, or about 19 or 20,000 genes. So you can see that more complexity in the organism, the more proteins they need for that organism to function. So they're going to need more DNA, a bigger genome, as well as more genes. I think I'm going to stop section 1, the first video there, except I just want to go back and point out that this one on simple gene expression, this little video that they have, is a good review. So it's not very long, and I recommend looking at that.